Hi everyone, thank you by advance for watching this presentation. My name is Théo Thréard. I am a third-year PhD student working at the Lima under the supervision of Professor Vitalyi Gusev, Dr. Samuel Raetz, and Dr. Nikolay Chigarev. My presentation today will introduce you to the method we have developed in order to go from the optoacoustic measurement of our sample to its 3D reconstruction on the computer. Let me introduce you to the TDBS technique. The technique we are using is named the time domain Brillouin scattering technique and has been developed by Humphrey Morris and his team in the 80s. In this technique, the pump laser beam generates an acoustic wave by thermoelasticity inside an optoacoustic transducer. This wave is then transmitted to the transparent sample, and the probe laser beam, delayed compared to the pump laser beam, is reflected strongly at the surface of the optoacoustic transducer, fixed, and is weakly reflected at the surface of the acoustic wave that is changing depending on the time and the position of the acoustic wave. These two reflections will interfere when collected at the photodiode. These interferences will lead to quasi-periodic oscillations named Brillouin oscillation, and the frequency of this oscillation are linked to the velocity of the acoustic wave propagating in the transparent sample by this formula at normal incidence. This means that when the acoustic wave will face some changes in the structure of the transparent sample, its velocity will be affected, and then the Brillouin oscillation frequency we will measure will change. Our goal is to recreate a 3D image of our sample from optoacoustic measurement. Here you have two examples of optoacoustic measurement realized on polycrystalline transparent material. Here you've got the example of a polycrystalline ceria ceramic material that was done by David Hurley's team in the US, and they were able to estimate the inclination between two differently oriented grains of the material. A second example of optoacoustic measurement realized in the loom on a high-pressure water ice has shown the inhomogeneity of the sample in its depth. This is a major information in our aim of imaging. This type of measurement have also been developed in the frame of imaging biological cells. In this first example, coming from Oliver's Wright team in Hokkaido University, they have been able to realize 3D imaging of endothelial cell in vitro. Finally, in this study from Bertrand Audouin's team in Bordeaux, they have been able to image cell structure as thin as 10 nanometers. However, to achieve this scanning precision, they had to develop an ultra-fast scanning system. Back in Le Mans, the experimental imaging system we are using has been developed by NETA. This ultra-fast imaging system is based on a pump probe technique, where a pump is used to generate wave inside the sample, while a probe laser beam is used to monitor the propagation of this wave. However, since the phenomena generated by the pump are ultra-fast, there are no electronic devices that can measure it directly. So, to overcome this limit, we are relying on the repetitivity of this phenomena in order to sample the measurement of the propagation of the wave using a delay between the probe laser beam and the pump laser beam. What makes NETA an ultra-fast imaging system can be found in the control of the delay. Instead of a mechanical delay line that would require a few seconds to do a single-shot measurement, the delay is controlled by the Aesop's principle. This means that the probe laser gravity is numerically enslaved by the pump laser gravity. Hence, in our case, one single-shot measurement can be realized in 2 milliseconds. To anticipate these changes, let's focus on the sample we are studying. Our sample is a polycrystalline ceria material with a gold nanolayer that plays the role of the optoacoustic transducer. This material has a cubic crystalline structure. This means that from the optic point of view, it is isotropic, Hence, the probe laser beam will not be refracted when facing a boundary between two differently oriented grains. However, from the point of view of the acoustic, this cubic crystalline structure means it is anisotropic. Hence, up to 3 quasi-acoustic modes can be generated in this kind of structure. Acoustical anisotropy means that the wave will not propagate at the same velocity depending on the orientation of the crystalline structure. Hence, to determine the Brillouin oscillation frequency interval, the slowness surfaces are plotted and the intervals are determined for each possible quasi-acoustic mode. The stronger anisotropy of shear waves is evident when looking to the 3D representation of the slowness surfaces. Since up to 3 acoustic modes can be monitored in this sample, a mode-by-mode signal processing technique has been developed. A scan of the Seria sample is realized with neta on a 60 × 60 micrometer square area with a 1 micrometer step in both X and Y directions. The first step of the signal processing aims to extract the acoustic component of the reflectivity signal measured with the photodiode. To do so, a bandpass filter is applied to the raw transient reflectivity signal in order to remove the noise. Also, the electronic peak influence, corresponding to the common time of pump and probe arrival, is removed since it does not have any acoustic information. Then, the thermal background is removed from the denoise transient reflectivity signal in order to extract the acoustic component of the signal. Applying a Fourier transform to the full acoustic signal gives information on which acoustic mode have been monitored or not. In the best possible cases, each of the 3 acoustic modes are monitored. This Fourier transform allows to extract the frequency of the Brillouin oscillation of each acoustic mode. And finally, if we regroup on the same representation all the contribution of the signals for a common mode, we can recreate a map of the Brillouin oscillation frequency depending on the position of the scan for one mode, here the longitudinal one. However, this information is averaged along the full path of the acoustic signal, and it does not give information on where does a frequency appear or disappear in the time and thus in the depth of the sample. To overcome this limit and study the frequency content of each acoustic signal over time, a short-time Fourier transform is applied. A Hanning window is slicing the signal over time, and the strongest frequency component inside the short-time Fourier transform for each window is found using a synchronous detection technique. If we regroup the Brillouin oscillation frequencies estimated in each signal for a same slice of the signal, we can have the same kind of representation as previously, but this time with an information over depth. Finally, gathering all together these slices of the sample allow a 4D stacked representation of our polycrystalline sample. This representation is composed of pixels located by their spatial coordinate x, y, z and by a color depicting their Brillouin oscillation frequency. Now, to shape these different grains that compose the polycrystal, we have extended Otsu's segmentation method in order to regroup the pixels or the point that belongs to the same grain. This cloud of points belonging to the same grain are then given as an input of an alpha-shape algorithm. This alpha-shape algorithm allows to shape a set of points using some constraint parameter. If the constraint parameter is null, then the set of points is shaped as itself, and if it's infinite, it is shaped as the convex field. And in between, you have all the possible alpha shapes of this set of points. In our case, if we use a tuned constraint parameter, we can obtain a 3D representation of the grains detected for each of the three possible acoustic modes. As I said, the processing is a mode-by-mode procedure. Thus, this explains why we have three types of results. What is really interesting here is the information given by the detection of the shear acoustic mode. What you can see on the 3D imaging results as well as on the first slice of the slicing procedure is that grains that were imaged as homogeneous when looking at the longitudinal acoustic mode, like the orange one or the violet one, are in fact composed of smaller grains when looking to the results of the shear acoustic waves. The enhancement of grain resolution by shear waves is due to the stronger anisotropy. These results are in very good agreement with the EBSD pattern that were done on the same scan area. These results highlight the key role of generating and detecting shear acoustic waves for a complete imaging of our samples. Finally, these 3D images open the possibility of geometrical estimates. This slide presents the study of an inclined interface between one grain and another grain that is imaged via two physical phenomena. The first one concerns the longitudinal acoustic mode. When the acoustic wave is transmitted from one grain to another, its velocity is changing. In that case, it is going faster. This means that the Brillouin oscillation frequency will increase. And if we report this depth of frequency shifting on a surface of frequency shifting, we can then estimate the inclination of this interface between the two grains. The second interesting physical phenomena concern a shear mode, the first one. In the case of this mode, the TDBS signal is disappearing. The same kind of surface can be obtained by reporting the endpoint of the time frequency representation, and the same kind of angle is obtained. In conclusion, we have developed a signal processing method in order to go from the optoacoustic measurement of our serial sample to the complete 3D representation of the grains of the area scanned. The imaging results allow to do some geometrical estimates, like the inclination between two grains. In the future, we would like to try to scan new areas of the sample for different geometries, try to enhance the spatial resolution in depth, and optimize the generation and detection of shear waves. Thank you very much for listening to my talk.